Very good morning to all the participants present here. At the outset, let me thank the organizers, the principal, the head of the department, Dr. A. Xavier, and the convener and coordinator, Mr. Vidya Sankar of the Michiro College, Madurai, for organizing this international webinar. In this webinar, it gives me great pleasure to share my research experience and knowledge in molecular modeling. Here we have Dr. T. Madasamy and G. Muniswaran, who are dedicated researcher, hardworking researcher, who have completed PhD under my guidance, now placed in UK and USA. They are also giving talks on solar-driven chemical fuel synthesis and molecular dynamics and simulations. My today's topic is molecular modeling and drug designing, an interdisciplinary approach. At the end of the session, you will be able to carry out your own computational theoretical calculation studies on your research interest for your final semester project or research project in your own area using the video tutorials presented here. What is molecular modeling? It is an important area of new area of research in multidisciplinary field discipline. Molecular modeling deals are used to construct and visualize the three-dimensional structures of drugs, biological substrates, to simulate and calculate using quantum mechanics or molecular mechanics and predict, analyze their molecular properties. It is also used to study the behavior of molecules at an atomic level. Molecular modeling is especially very useful for highly explosive compounds, unstable compounds, which cannot be studied. Their properties cannot be studied. For example, heat of combustion, heat of formation, those cannot be computed, determined by experimental method because of their instability, explosive nature. In such cases, molecular modeling is a valuable tool to analyze their properties. In recent decades, molecular modeling is expanded. It also covers data mining to organize many drug compounds and their properties into database. It is also used to perform molecular docking and virtual drug screening for drug discovery. What are the applications of molecular modeling? Molecular modeling has got lot of applications in several disciplines, in biology, in chemistry, in physics, etc. It is nothing but a set of tools, techniques for studying the chemical and physical properties of a system, of a molecule, new molecule or drug using a computer. For example, 3D structure can be drawn and its molecular geometry could be determined. Electron density distribution, how the electron density is distributed at different positions of the molecule, thereby we can predict the chemical reactivity and the mechanism. We can calculate the IR spectrum and compare with the experimental IR spectrum and also UV visible spectrum. We can calculate the frontier orbital energies for pericyclic reactions, for the calculation of redox potential, circular dichroism spectra, ESR spectra, NMR spectra, Mossmeyer spectra. All the spectra could be calculated, theoretically computed, simulated. Thermodynamic properties such as heat of combustion, heat of formation, delta G, change in free energies, redox potential could also be calculated. The reaction mechanism can be modeled and compared with the experimental data. We will see now the interaction mechanism of drug with an enzyme, inhibitor, physical properties such as nonlinear optical properties. It is valuable for drug designing, protein modeling, and molecular dynamics and simulations. We'll see one by one. I will also be giving video tutorials for you to carry out for your research problems. Molecular modeling is an interdisciplinary field. We need the knowledge of chemistry, biology, computer science, physics, maths to understand fully the molecular modeling. We also need the expertise of mathematician for developing algorithm, simplest algorithm to calculate very fast the calculations. Biologist for the sequencing of proteins, identification of proteins, genes, DNA. Computer scientists are required to write a program for the algorithm, and we need the physical properties of the protein molecule like Ramachandran plot. We also need to know the chemical principles involved in in developing the molecular modeling softwares. There are four types of fields. They are disciplinary field, multidisciplinary, interdisciplinary, transdisciplinary. In disciplinary, we will focus only on one subject, for example, chemistry only or physics only. In multidisciplinary, it is a two major system. We will study both chemistry and physics, or both are major. Interdisciplinary, for example, biochemistry, they work at the interface of chemistry and biology. Transdisciplinary, it is the integration of several disciplines to get a new novel invention by the integration of several disciplines. We'll see later. Nowadays, interdisciplinary research is promoted by Department of Biotechnology, Department of Science and Technology, SERB, all the funding agencies. What is interdisciplinary field? Interdisciplinary field is research, teaching, or problem solving that integrates several disciplines to create a new, unified, novel outcome or invention, according to James Collins. In recent days, you will be given grant only if the research project involves interdisciplinary aspects. Why interdisciplinary, multidisciplinary aspects research are promoted? Because it accelerates the research progress because of the involvement of experts from several disciplines who can contribute to solve a problem, and it also expedites the transition of results to application. It also prepares students to work in different disciplines and also work with the several discipline experts. There are several interdisciplinary fields, for example, molecular modeling, drug designing, computational chemistry. It is also computational chemistry or computational physics or computational biology, nanoscience, nanotechnology, neuroscience, neurochemistry, biosensors, biophysics, bioinformatics, etc. I worked in Medical College of Wisconsin for the last 5 years. Medical College of Wisconsin is a part of Frydet Hospital with the helipad and Children's Hospital, one of the biggest hospital in Wisconsin. In Medical College of Wisconsin, I work for Biophysics department. It consists of two divisions, EPR and NMR. In National Biomedical EPR Center in Biophysics department, there are experts from different disciplines. For example, electronics, instrumentation engineer, physicist, chemist, biologist, medical doctors, all are working towards solving a particular chemical biomedical problems. Let us see the principles of molecular modeling or computer aided drug designing. They are based on molecular mechanics and quantum mechanics. Molecular mechanics is nothing but classical mechanics, and quantum mechanics is nothing but for applicable for small molecules, whereas molecular mechanics is applicable for protein molecules, DNA, macromolecules. Quantum theoretical calculations are based on quantum mechanics, quantum theory. There are well known theoretical methods such as ab initio, Hartree-Fock, density functional, semi empirical methods. These quantum mechanical methods uses well known physical constants only, such as velocity of light, masses of electrons, nucleons. These are only used to calculate the observable molecular properties, any observable molecular properties. This is calculated using the Schrodinger wave equation, which you have studied in quantum mechanics class. It is nothing but eigenvalue equation A psi equal to E psi. For example, in the Schrodinger wave equation, H cap is the total energy operator, is called Hamiltonian operator. It is the sum of kinetic energy and potential energy. The Hamiltonian operator for hydrogen atom, for one dimensional box, for molecules can be easily constructed mathematically. So once you know the operator and the wave function, by assuming the hydrogen atom approximate wave function, and we can calculate the observable values. For example, here energy or any other observable properties. Okay, what is eigenvalue equation? When the Hamiltonian operator or when any operator of corresponding to a particular physically observable property operates on a wave function, it gives the observable values, in this case, observable molecular energies and the wave function. The wave function is nothing but, the wave functions are nothing but your atomic orbitals. They are used to describe the state of the system or describe the molecules. So this is the fundamental equation used for the molecular mechanics, quantum mechanical calculations to obtain the various observable molecular properties. In this, let us apply the molecular modeling to understand and investigate the Parkinson disease. What is Parkinson disease? It is a neurodegenerative disease affecting the central nervous system. Especially, it occurs due to the loss of dopamine. Dopamine is an important neurotransmitter present in dopaminergic neuronal cells. In Parkinson disease, it affects the motor skills, speech, and other functions. Especially, it causes tremor, shaking of hands, especially at the old age. After 60, everybody will have Parkinson disease, and also drug addicts, they will get the disease at their early age. There is no medicine for this disease. As I said earlier, there is no effective drug for the treatment of Parkinson's disease, it is very much necessary to investigate the mechanism of the Parkinson's disease. In this, we have carried out the theoretical investigation of metabolites of dopamine quinone, with the formation of ADDUCTS with the DNA nucleic acid bases. Dopamine is an important neurotransmitter, is formed in the neurons of the substantia nigra of brain. Under oxidative stress condition, especially at old age above 60, all of them will have minor Parkinson disease symptoms because of oxidative stress. So it is necessary to take antioxidants rich fruits to detoxify, to deactivate the free radicals and oxidants generated at old age. Similarly, drug addicts are also affected by Parkinson's disease, tremoring, shaking of hands, because of at a younger age, because of this loss of dopamine. So, under oxidative stress, dopamine is oxidized to dopamine quinone. This dopamine quinone then forms adduct with the nucleic acid bases such as adenine, guanine. They are electron rich in DNA, causing DNA damage, leading to carcinogenicity. This dopamine quinone is very, very reactive, so it forms immediately adduct with the DNA bases. It is experimentally found that the dopamine quinone, the metabolite of dopamine, forms adduct with the adenine at N3 position and N7 position of guanine. The structures are shown here. Using molecular modeling, we have considered the various possible attacks of dopamine quinone on these quinone, guanine, and adenine by following the activation barriers. From the activation barriers, N7 attack and N3 attack of adenine are having the lowest activation barriers, indicating the formation of the adducts formation with the N7 and N3 position of adenine and N7 position of guanine. Here I have shown the optimized geometries of the DMQ adduct with the nucleic acid bases guanine and adenine are shown here. Our theoretical modeling illustrate that DMQ forms stable adducts at the N3, N7 positions and N3 positions of guanine and adenine respectively. We have further analyzed the geometry of these DMQ adducts with the base pairs. It reveals that the DMQ causes remarkable changes in the structural, conformational, and hydrogen bonding properties, leading to mutagenic effects such as base mispairing. It also explains the toxicological behavior of DMQ, why it is neurotoxic, why dopamine is neurotoxic under severe oxidative stress condition, why it is carcinogenic. We have published the data in the Journal of Structural Biology on the theoretical investigation of quinone metabolites of dopamine adduct with the DNA, insights into toxicological effects. As you are all advised, don't eat fried foods. Why fried foods are dangerous? Fried foods are even poisonous because acrylamide is a potent carcinogenic material to animals and humans, is reportedly to be present in a very large number of human consumed foods, especially fried foods, at the level of milligram per kilogram. So here we have used why the acrylamide is carcinogenic. We have studied the mechanism through theoretical modeling, theoretical calculation. As I said, acrylamide is a carcinogenic material. It undergoes epoxidation in the presence of cytochrome enzymes present in liver. It is produced in the liver. It oxidizes um, acrylamide to give uh, glycidamide epoxide derivative. This glycidamide is very very reactive species as like DMQ. It again forms attack with the nucleic acid bases such as guanine, adenine. So we have modeled again the reaction of the formation of adduct of glycidamide with the nucleic acid bases. Glycidamide, a metabolic product of acrylamide formed via epoxidation, has been proved to be responsible for the carcinogenic behavior. So why it is carcinogenic? Why how it is carcinogenic? We have carried out this investigated the possible mechanism by which the reactions of glycidamide, metabolite of acrylamide with DNA. Using molecular modeling tools, theoretical modeling, we have calculated, we have modeled the mechanism of, for example, we have shown here N7 attack on glycidamide to form N7 attack through this intermediate. We have calculated the activation barrier for N7 attack and all the possible attacks, and found that the activation barriers for the N7 adduct is lower. Therefore, the N7 adduct is preferentially formed, as you will see. Our theoretical investigation by computing the activation barriers and analyzing the thermodynamic parameters for the attack of glycidamide on three different sites. We have considered N3, N7 and O alkylation of guanine in the GC pair. It showed that N7 is the preferred position in agreement with the experimental data reported. The formation of glycidamide adduct with the guanine GC pair caused remarkable drastic structural changes in DNA, especially major and minor grooves, leading to its mutagenic effect and carcinogenic behavior. Thus, our studies provided insight into the mechanism of carcinogenic properties and act as a useful guide for designing new drugs in the future. The above work carried out is published in Computational and Theoretical Chemistry journal. Theoretical description of cytotoxic potential of glycidamide, an epoxide metabolite of acrylamide. What are the softwares for molecular modeling, especially theoretical and computational calculations? There are several softwares in the net. Some of them are free, some of them are licensed. For example, Gaussian, we have used here, GaussView, we have used here extensively for 3D structure drawing, editor, analysis, display, and for theoretical calculation. There are other softwares like Chemists US, Chemists UK. These are all. This is a free software. We have used Hypercam, Orca. It is a free software. Mopac, Schrodinger. It is a licensed software. There are several structure. You can construct the 3D structure of the drug molecule using Gauss Weave, Gab Edit, Avogadro, Argus Lab, Chem3D, etc. For theoretical calculation of IR spectra, UV visible spectra, or NMR, ESR, HOMO, LUMO, frontier orbitals, there are four steps involved. Basically, there are four steps involved. Step one, it involves 3D structure construction, drawing, and building the molecule, building the drug molecule. Then, in step two, the structure is optimized. In step 3, the UV visible IR or Raman or any mass Bayer spectrum is calculated, and in step 4, it is plotted and analysis of HOMO LUMO. For example, in this video, we will demonstrate the theoretical calculation of IR, UV visible, HOMO LUMO. So far, we have studied the molecular modeling of small molecules. In this, we will study the modeling of protein, DNA, or macromolecules and their dynamics. For example, X-ray crystallography gives the static structure, the three-dimensional coordinates of each atom in the protein molecule. As I said, there are two types of methods: quantum mechanical methods and classical mechanics, molecular mechanics method. We have seen quantum mechanical methods based on quantum mechanics are applicable only for small molecules, especially 10 to 100 atoms, and molecular dynamics 10 to 100 picosecond. Whereas classical mechanics based on Newtonian mechanics, F equal to ma, we can apply for complex proteins, enzymes, DNA, RNA, etc. It is useful for calculation of molecular dynamics, molecular modeling of atoms, 10 power 4 to 10 power 5 atoms, and we can study molecular dynamics up to 10 to 100 nanosecond. This classical molecular mechanics or classical mechanics is based on mathematical model of a molecule as a collection of balls. In the case of classical mechanics, molecular mechanics, proteins are considered as a molecule, as a collection of atoms are considered as balls. They are held together by springs. In molecular mechanics, the energy of a molecule is a function of its bond stretching, bond bending terms, dihedral terms, van der Waals terms, and electrostatic terms, where B is the bond length and B equilibrium is the equilibrium bond length, theta is the bond angle, and theta equilibrium is the bond angle at equilibrium, dihedral angle phi is the dihedral angle, and R is the distance between two atoms in a protein molecule. These parameters Kb, K theta, Vn are form called force field parameters. These parameters are very useful in the calculation of molecular dynamics and molecular simulations of especially large molecules, proteins, DNA, etc. Now let us apply the molecular mechanics, molecular dynamics, molecular simulations method to understand the neurodegenerative disease, amyotrophic lateral sclerosis, ALS disease. It is again a neurodegenerative disease similar to Parkinson's disease. It is also called Lou Gehrig disease. It is a fatal This is caused by the degeneration of motor neurons in the brain. For example, Stephen Hawking was suffered due to this disease and died of this disease. He was at the age of 21 years due to this ALS disease, due to paralytic attack. This is paralytic attack disease. Similarly, Lou Gehrig's, a baseball player in USA, he died at young age, all of a sudden because of this ALS disease, neurodegenerative disease. Again, there is no cure for no cure, no effective drug for neurodegenerative diseases. Let us see now what is the cause of this paralytic attack disease, ALS disease. What is the mechanism of this disease? Again, we have used the molecular dynamics, molecular modeling, simulations methods to understand the mechanism of the disease. There are there are two types of ALS disease. Familial ALS disease due to mutations in copper zinc superoxide dismutase enzyme, and another is due to sporadic ALS. It is due to oxidative damage of SOD1, cytochrome C, and other proteins. Especially, as I said, after 60 years old, you are you will be you are under oxidative stress. You'll be producing lot of free radicals, which causes extensive damage to the cell organelles, cell proteins, etc. It leads to uh, paralytic attack or mild form of ALS disease. What is mutation? Due to mutation, ALS disease occurs. There are more than 90 mutations are reported in the literature. What is mutation? Mutation is nothing but one of the amino acid in the polypeptide chain of the SOD1 enzyme is replaced by another enzyme, another amino acid residue. For example, alanine is replaced by valine at fourth position in the protein sequence. Similarly, at 93rd position, guanine is replaced by alanine. So there are some of the mutations, for example, you have a sentence. If you if you replace one letter by another letter, it will change the meaning. You will change the meaning. For example, die, dye, die, die. So such mistakes also occur in our body, human cells. Such replacement of one amino acid by another is called mutation. This mutation, due to these mutations, ALS disease occurs, and many more disease occurs due to this mutation. As we make mistakes in writing sentence, it is misunderstood by other people. Let us see another example of mutation leading to sickle cell anemia disease. This is the normal sequence. This is the normal sequence. I have shown the partial sequence region of DNA, and DNA is translated into protein, proline. This triplet codon is translated into proline, and this is to glutamate. In the case of single point mutation, glutamate is replaced by valine because of mutant gene, mutation in gene, and therefore single point mutation in sickle cell anemia disease patient. This is the red blood cell. It is globular, whereas in the case of sickle cell anemia, there's the conformation changes. It is sticky, and not it is not globular. By single mutation, you have 300 amino acids. By just replacing one amino acid by another, it causes a lot of changes in the three-dimensional changes, structure of the protein and the dynamics of the protein. Therefore, the functions of the proteins are also affected because of the single point mutation, leading to sickle cell anemia. Why the mutation in SOD1 enzyme linked to ALS disease? For example, this mice is G93A mice is paralytically attacked after 9 months old because of SOD1 mutant enzyme is expressed. They are very toxic to motor neurons. So we have studied in Medical College of Wisconsin why, whether it is what is the toxicity of this enzyme. Does mutant SOD1 have less activity? We have measured the activity of G93A and found that the activity is not lost. Superoxide dismutase activity is not lost, whereas they have gained other activities such as peroxidase activities, thiol oxidase activities. We have used, we have identified, we have determined these peroxidase, thiol oxidase activities of the mutant enzyme using ESR spin trapping method and low temperature EPR and advanced EPR technique. So we have applied the molecular dynamics, molecular protein modeling simulation method to understand the effect of mutation on copper zinc superoxide dismutase on the dynamic properties and and thereby the activities changes. In this, we have studied the structural conformational changes on on the mutations of copper zinc superoxide dismutase by molecular simulation method. As I said, mutations in SOD1 is linked to familial amyotrophic lateral sclerosis, a, a fatal neurodegenerative disease causing paralysis and death. It is occurring mainly due to folding, protein misfolding, conformational changes, aggregation, resulting in the enhanced peroxidase and thiol oxidase activity of these mutants. Here we have carried out 20 nanosecond molecular dynamics simulation for wild type protein and H43R and tryptophan is replaced by phenylalanine at 32 position mutated SOD1's dimer. We have compared their structure, conformational properties from their trajectories to understand the pathology of the disease. Our results showed that there is increasing distance between the critical residues, thus resulting in the open conformation at the active site. Especially the active site of copper zinc SOD is very narrow. It allows only superoxide and reacts at that copper site, whereas in the case of mutation there are a lot of structural conformational changes resulting in open conformation, resulting in the peroxidase activities. Here I have given the wild type SOD1 structure and mutant protein, mutant SOD1 structures after 20-second molecular dynamic simulations. As you see, there are changes in the conformations of the mutants compared to the wild type protein. Let us now see the molecular dynamics video of wild type SOD1 and the mutant SOD1 in the following video. Also, I have given the video tutorials for how to carry out the theoretical calculations, how to carry out molecular dynamic simulations, how to mutate the protein obtained from protein database bank, and molecular docking, how to dock cyclodextrin with the drugs, how to dock the protein HIV protease with its drug, etc. Please watch in the afternoon session. Here we have studied the molecular dynamics of cytochrome C, especially cytochrome C oxidized at tyrosine 67. Cytochrome C is involved in electron transport chain in mitochondria. It transfers electrons from complex 3 cytochrome C reductase to cytochrome C oxidase. While oxidizing this residue, it is not able to transfer subtle electron between complex 3 and complex 4. Because of oxidative stress, due to oxidative stress, um, tyrosine is oxidized to nitrotyrosine. As a result, there is a conformational changes, structural changes, and activity changes, especially because of the tyrosine 67 at the active site. Using molecular dynamics simulation, we have studied the structural and conformational changes in tyrosine 67 nitrated cytochrome C protein and published the work in molecular simulation. What are the softwares used for protein modeling, molecular dynamics simulations? For molecular dynamics and simulation, there are several softwares, well-known softwares: CHORM, Ampere, Schrodinger, Hyperchem, Chromax. In this case, we have used Chromax for molecular dynamic simulation. It is available free. Chromos, NAMD, VMD, it is also free available. We have used this software again. For virtual screening, Raccoon 2 is used, we'll see later. And for visualization of proteins, analysis of molecular dynamics data, we have used RASMOL, PyMol, JMol, Chimera. Chimera is used to cause mutation. For example, copper zinc SOD, we have mutated the protein by using VMD, Chimera, Swiss PDB, we have used these softwares to analyze the data, and also for mutations and hydrogen bonding analysis, etc. In this slide, we'll see how molecular dynamics simulations were carried out. There are essentially seven steps involved in molecular dynamics simulation. In the first step, the PDP data is retrieved, is obtained from the PDP database. It is available free. You can get the PDP data for SOD1, Cytochrome C, or any protein whose X-ray crystal structures were already reported and deposited in protein database. We can, using Chromax, we can generate topology and define box and solve it, add ions to make it neutral, and then it is energy minimized. The whole system is energy minimized and equilibrated, and then molecular dynamic simulation is carried out for 10 nanosecond or 20 nanosecond. As you see, the longer the longer the dynamics time, you will get the more accurate dynamics of the protein and their dynamic function relation, and then analysis and results and discussion. In this, we will study about drug designing and discovery. It is very, very important. For example, coronavirus, how drugs are designed for HIV or coronavirus or dengue virus. Let us now see the steps involved in drug discovery and development. For example, a person is suffering from fever and is the virus or the bacteria causing disease is isolated by microbiologists, and using genomics or proteomics, the various genes expressed and the various proteins expressed by the virus, for example coronavirus, are identified. The proteins are present in large concentrations, are isolated, and they are isolated as potential target protein to inhibit the protein activity and thereby killing the virus. Virtual screening of drugs using molecular modeling is carried out. We will see one by one using drug database and followed by high throughput screening. From the virtual screening of drugs, a drug with good inhibition, good binding at the active site is chosen, and it is preclinically tested. Testing is carried out. Let us briefly see genomics or proteomics. In genomics, we look for mRNA expressed, genes expressed. In proteomics, we look for proteins essential for infectious agent distinct from host genes, and proteins are identified, and then find regulatory pathways required for disease process. After the drug preclinical testing for 1 to 3 years, the drug is formulated and it is scaled up, the production is scaled up, and human clinical trials will be carried out for around 2 to 10 years, and then FDA approval. It will take another 2 to 3 years, then it is released into market for the cure of the disease. In the last slide, we have seen virtual screening of drugs database. It is carried out using molecular docking. So, what is molecular docking? Why it is important? For example, molecular docking is used for virtual screening of drugs and drug designing. Molecular docking is based on shape complementary interactions which attempt to find the best match between the protein and the drug. Let us see now what is computer aided drug designing, CAD. There are two possible ways one can design the drug. For example, protein structure where the active site the drug is bound ligand based from the substrate biological substrate based. We can design the inhibitor using QSAR is quantitative structure activity relationship. It is a mathematical relation, gives the relation between the molecular properties, physicochemical parameters of the drug molecules, and the inhibition activity. Whereas in the case of protein structure based, we need to know the crystal structure of the protein. If the crystal structure of the protein is not known, only the sequence of the protein is reported, then using homology modeling, we can determine we can model the structure of protein from the sequence already reported, which best matches with the unknown protein structure. The structure of the protein is determined, we'll see further. And then computational analysis of molecular docking and determination of free energy of binding between the protein and drugs. What are the advantages of CAD virtual screening? It saves the time, cost, and gives a lot of information about the disease, bioinformatics, and the screening time is reduced. If you do experiments, it will take long time. Initial database, initial screening is carried out, and then the experiment is carried out to confirm the study. In this, we need less manpower. CAD molecular docking is very important. How docking is carried out? We'll see now. Docking, the process by which molecular modeling software fits a drug molecule into the active site of the target protein molecule. Docking is frequently used to predict the binding modes, binding orientations of the drug molecules in the active site of the protein target. It also predicts the affinity, free energy of binding, and inhibition mechanism of the drug molecule. So, docking plays an important role in the rational design of drugs. There are three important types of docking: protein-protein docking, protein-protein docking, protein drug inhibitor docking. For example, HIV protease is identified and inhibitor docking, small molecule docking. For example, cyclotextrin drug docking. Cyclotextrin is used for drug delivery and chromatographic separation of enantiomers, and also in sensing of biological substrates. What is inclusion complex? For example, cyclotextrin, when it forms complex on docking with the drug, It forms inclusion complex. The internal cyclotextrine is this, and the drug is docked here, forming an inclusion complex. The internal cavity is hydrophobic, therefore, insoluble drugs can be trapped into the cavity of the beta cyclotextrine, and it can be encapsulation of drug for useful for drug delivery. What is beta cyclotextrine? It is nothing but a cyclic organic compound derived from starch transformation through enzymatic process. Beta cyclotextrine consists of seven glucopyranose units. Alpha cyclotextrine consists of 6, and gamma consists of 8 glucopyranose units. Here I have shown the beta cyclotextrine aspirin inclusion complex. For molecular docking studies, we need to know the three-dimensional structures of proteins, target protein. The three-dimensional structures of proteins were determined by X-ray crystallography or NMR spectroscopy or homologous modeling. What is homology modeling? In homology modeling, get the amino acid sequence of the target protein. For example, coronavirus, you have identified the target protein to inhibit the protein or enzyme. Get the amino acid sequence. Align the above sequence of target protein with the known protein sequence as a template. Using modeler Swiss model or Swiss model softwares, we can determine the three-dimensional structure of the target protein from the amino acid sequence using these softwares. This is the schematics of molecular docking process. To docking program protein structure, 3D structure plus drug database are given as input, and the success of docking program depends on the algorithm and the scoring function we will see. There are two types of docking: rigid molecular docking. In rigid molecular docking, the protein and the ligand, the drug has rigid bodies. It neglects the conformational degrees of freedom of ligands. In flexible docking, we will consider the different conformations of the drug. There are two types of docking methods based on shape complementarity and complementarity in bonding and non-bonding interactions. In shape complementarity method uses a matching technique that describes the protein and ligand as complementary surfaces, whereas in the second approach it simulates the actual docking process, and the drug protein pairwise interaction energies are calculated using scoring function. You will see both approaches have significant advantages as well as limitations. In molecular docking, the scoring function is very important. It calculates the free energy of binding from the from the summation of hydrophobic interactions, hydrogen bonding interactions, ligand orientation with the best complementarity score, binding affinities, ionic interaction, aromatic interaction, van der Waals interactions, electrostatic interactions. Now we will see what are the docking softwares available For example, doc is one of the software. In our case, we have used Autodoc. It is available free. We have used for molecular docking and screening, and also to calculate the free energy of binding between the protein and the drugs. Similarly, there are other softwares, Flex, Gold, developed by UK. In this, I will show you how rational drug designing is used for finding drugs for HIV protease. We will see how drug designing and drug discovery is applied to HIV disease. For example, a person is identified with the HIV disease. The virus is isolated and identified by the microbiologist, and biologist and biochemist using genomics and proteomics. They will find out the genes overexpressed and HIV and HIV protein. They have identified the protein as a particular target for this HIV virus, and X-ray crystallography, using X-ray crystallography, NMR spectroscopy, the structures or homology modeling, the structures of HIV protease were obtained by physicists and chemists, and then structure-based drug designing is carried out using molecular docking and virtual screening. Once the crystal structure is known for the target protein, for example, HIV protease, using molecular modeling, molecular docking, computer aided drug designing by computational chemist, we can we can find out the effective drug by means of docking and molecular dynamics simulation, and further experimental assay studies were carried out. By molecular docking and virtual screening method, HIV drug Intinavir was reported and developed and used as drug, where it binds at the active site, tightly at the active site, resulting in the inactivation of the protein and thereby killing the virus. Over a time, the drug is found to be inactive. Why the drug is found to be inactive? Earlier, the drug was very effective, and the persons were cured. Nowadays, they are not cured because of they evolve a mechanism. The virus evolve a mechanism whereby they take different route. Similarly, there are several mutations reported in the HIV virus, especially in HIV protease, where the drug is not tightly bound because of these mutations. As a result of this, the drug is not very effective, and this drug is inactive. It is ineffective. Such mutations are called drug resistant mutations, or were reported. In such above cases, we can carry out the mutation using computational tools, Swiss PDP, Weaver or VMD. We can mutate the protein by one amino acid by another amino acid, and we can carry out molecular docking and determine the binding energy by using autodock. If there is a decrease in binding energy, then due to mutation, then the drug is ineffective. We can, instead of doing site directed mutagenesis, which is very very tedious process, genetic engineering it involves, and overprotection of enzyme, it is very very, protein is very very tedious, complex, long process. By using protein modeling, molecular modeling, we can easily carry out and check the whether the enzyme is whether the protein is binding or not, by affecting the mutations using computational tools. In now, what are the steps involved in molecular docking using AutoDock? We will demonstrate and give you the video tutorial as follows. The first step is preparation of drug. Using molecular modeling tool, we can draw the structure and optimize the structure and get the coordinates for docking. Preparation of target protein, the PDP data bank, PDP data bank, we can get the PDP file, three-dimensional structure data file. Then grid map optimization is carried out using AutoGrid 4, and then the docking parameters were selected. We will show you, and using AutoDock 4, docking is molecular docking is carried out, and further analysis is carried out using Autodoc tools available. It is freely available. We can get the binding orientation and the free energy of binding of the inhibitor with the target protein. As I said earlier, QSR deals with the ligand based drug designing. What is QSR? Quantitative structure activity relationship. It is a mathematical relation which gives the relation between the activity, inhibitory activity, and the physicochemical parameters of the molecules, drug molecules. There are several well-known softwares to calculate the QSR parameters as follows. These are our publications in molecular modeling, dynamic simulations, in Journal of Molecular Graphics, Molecular Modeling, Journal of Informatics in Medicine Unlocked, in Biophysical Journal, in Journal of Structural Biology, and in Journal of Computational Theoretical Chemistry, in molecular simulation, in sensor letter for theoretical screening and selection of functional monomers for molecular imprinted electropolymer as sensor for glutamate. We are very thankful to Gaussian, Cosweave softwares and other authors who have contributed to this field. I am very much thankful and grateful to Dr. K. Muthukumar, USA, and Medical College of Wisconsin, where I worked for 5 years. Mr. M. Karthikeyan, Dinesh Kumar, and Kaniraja, Research Fellow, Biomedical Research Lab. They have developed the tutorials for you and other research, former research fellows who carried out research in our lab, who contributed in this field. Finally, I thank all my research scholars, former research scholars and present research scholars for their support and for their hard works. They are now in USA, Thank you all the participants for patiently hearing and watching this session. Please continue watching the video tutorials in the afternoon session for how to carry out the molecular dynamics, molecular modeling, molecular docking, and drug designing calculations. Thank you for watching.